So our next speaker is the CEO of Ultragenyx, Dr. Emil Kakkis. And one thing that I think we all should know about Emil is that he really started his career in drug development, I guess I should say. He wanted to be a vet. I should say that first. He wanted to be a vet, and he wanted to do a DVM PhD, and then someone advised him that if he really wanted to get into research and medicine, that it would be better to go to human medicine. And so I think they guided him well, and I should say they didn't, because I'm a vet and we'd be better off being vets together. But considering what he's doing for Angelman syndrome, I'm really happy that you're not a vet. I'm really happy that you're an MD. But ultimately, he started his career with a single patient and a single family. And that really got him started in drug development, and that was saving Ryan. And Ryan had MPS 1, and I'm sure we've heard his story about Ryan before, but those of you who haven't, it's an amazing story that you should read. He wrote a book about it, actually. And it really speaks to me and all of us personally, because we as families reach out to researchers, and we called Jim Wilson, we called Scott Dindo, we called David Siegel, we called Ed Weber and said, this is amazing work that you're doing, can you do this for my child? And you did it for Ryan, and then you've now done it for thousands of other kids around the world, and hopefully for Angelman syndrome now as well. So we thank you for that. And it all starts with one child and one disease, and hopefully grows to many, many more, and we're doing that for Angelman and hopefully many neurodevelopmental disorders. So with that, Dr. Emil Kakkis, CEO of Ultragenyx, is going to be speaking us today about the development of rare disease therapeutics, compassion and transparency. Thank you.
Thank you, Allison. Allison was one of the persons that called a number of years ago when she first found out about her child having Angelman syndrome. So I'm happy to be here today. I think this is the third talk I'll give. And as part of my general theme, I've always been talking about philosophical issues about doing rare disease drug development, and that will be today as well. I decided to talk about compassion and transparency as the beginning of the talk, just the philosophical differences of doing clinical trials in rare diseases from what are pretty traditional ways of doing drug development, and our learnings over the time and how it can be done well and managed. And then I'll transition and because Allison had asked for it, some review of our existing ASO data and where we're at in the beginning of that program for GTX 102. So, in drug development, there's a lot of sort of history of how it's done in a lot of the common diseases, but right now it's really, as Jim was mentioning, a golden age really for rare diseases, because all these treatments and all these new modes are coming forward. We're actually taking on diseases in credible ways that will potentially change the future. And these are specific treatments, not just symptomatic treatments, and they're a new technology, but there's a lot unknown, and drug developers and investigators and patients are going to have to collaborate to figure this out. And the truth about this is that drug development in these kind of diseases, complex, rare, devastating disorders, requires a deeper connection and more transparency and connection regarding how to do this, because of some things I'll explain to you now, but I think it's just very different from other types of diseases, like type 2 diabetes or blood pressure or all these other common diseases for which the paradigm of drug development has been created. And through my learnings over time, I've realized you have to handle this differently, and for a number of reasons, which I'll explain. So what really is so different about rare diseases? Well, first off, the disease impact is incredibly large, right? These are things that affect them for their entire life. It is by far the most dominant thing, not only in their life, but frankly, every single day. It's just so much greater than a lot of other diseases that get for drugs being developed. I think that's one of the key things. They're also complex, less understood, so we are constantly struggling to figure out what's really going on and what can we do. And the drugs are highly specific, addressing very sophisticated mechanisms, and therefore we're doing things that haven't been done before. This creates a lot of uncertainty. This is where there's a need for a closer connection and transparency and compassion, or how you do this. Now, what is a study participant? What does it mean to them to be in a trial? For for these type of diseases? Well, it's by far, after the diagnosis, the most important thing in their life is being involved in a trial. It's a fundamental issue. Is this going to lead forward? I mean, it's going to change my future or not? So this is not a subtle thing. It's not a minor thing. It is a deep and important issue. And one thing we know in these trials is that no matter how smart you are, no matter how much work you do, when you're doing the trial, you will figure out something that's not right, that you're going to have to fix, you're going to have to adapt. I talked last time about, the things that happen in trials and the fact that you have to be able to do that. So you have to see what's going on. And I say this because many times people talk about doing blinded, randomized trials in patients for the first time. It is very hard and rare to pick everything correctly and to know what to do. There's nothing to do about knowledge. There is no knowledge when you think you know from a natural history a disease, but you don't. You don't really know a disease until you start treating it. When you start treating disease, that's when you learn about the disease. The natural history is sort of watching at a distance. It's by putting a drug into someone and seeing the reaction that starts to teach you what's really going on. And when you do that, things happen, and you have to manage it. And this is why, even though the FDA will often recommend it, other people from an ivory tower perspective like the purity and beauty of randomized early studies, the truth is you won't be able to solve those problems on the fly. You won't see it coming. You won't be able to handle it because no one knows who's on treatment until the thing gets unblinded. So as those things are brewing, you will not be able to make good decisions. So this means it's hard to do those kind of studies in these patients. You have to think about how to do this while knowing a patient's getting the drug, how to do it smart and objectively. So there's two things that come out of this, which is you need to manage development differently in rare diseases. And if you want to develop a good, connected, warm relationship with families as you struggle together to get to the truth, one of the things that things will happen during development, and compassion use has always been a problem for industry, and it shouldn't be, and we need to engage. And I'll talk a little about that as a second topic, because things will happen in the patients you're treating or in other patients. And how do you look through that? How do you manage that situation when what you're doing is the most important thing for a disease, anywhere in the world? The transparency part is the tricky part. Patients are putting their lives at risk, and should they know what's happening? Should they know what's going on? There's a view that no, we're not supposed to tell them to try to keep it clean and objective, but it is a very difficult thing to completely block any knowledge and try to control it when this is so important to their lives because of the reasons I've described. So the objectivity question versus transparency is a debate. Somewhere in between not telling them anything and telling them, sending social media texts moment to moment when results come out, somewhere between those two, there's a place where we can give people a reasonable connection to what's going on when we know it, while at the same time working hard in a collaborative way to develop the right objective assessments. And that I think you can do it in a way that allows you to predict whether what you're seeing is true or representative, and that you can predict doing an appropriate randomized trial eventually. Now, my own experience is I put these 6 programs, but all of these 6 programs, we did open label trials. We did tell patients what was going on in the trial. We were able to get objective insights on how the drug was working. We're able to do randomized trials and demonstrate the effect. So the idea that you can't do that is just not true. In fact, most recently we had some randomized placebo-controlled phase 2 trials as a first trial, and we've just retracted that and went back to open label because, in fact, it doesn't work well. We just did that for 2 studies. So what I'll tell you is that while it looks pretty and beautiful on the whiteboard, in the real world, it's hard to do. We've done it, though. We've taken open label trials and gotten through and done it, and I think it's a better way to approach rare disease drug development. So being objective in open label trials is possible. We've done it. The rare disease patients, they need to discover what this is doing, and they need to be part of that process. And the paternalistic view that say, no, you're a patient, you sit out there and wait for us to tell you what's going on, it's just not the way things are. Rare disease patients and families today are deeply connected to what's going on in the world. They are highly educated about their disease, often more than the investigators. They need to be engaged, and learning and adapting is enabled, and when they're connected with you, then you can actually do a better job. When we are doing this objectively, we also need to be looking at what we're doing. Dose and dose administration could change. Changing how we're managing infusions, how we're managing other things. There's a lot of places, and I'll show you some examples of these. And so we know when we're working with them, the investigator and the patient should be saying, look, we're working to get objectivity. And I've watched patients and families for many years. I guarantee they're not looking for false hope. They're looking for real evidence of change in their kid. That's what you want to see. They're not trying to make up stories just because they feel better. Most of the patients and families have been through a lot already. They've gotten well past the levels of denial and other things. They're at the point of understanding, I'm ready to get on, especially families that enter trials are interested in figuring out what's going on. So let's work on getting it objective. Let's look for the evidence that gives us confidence that this is real. So examples, first program I did was Aldurizam, and I was the investigator, not the company at that time, and I had 10 families to deal with. And I had to come up with, what do I do about telling them what's going on? And I decided it's impossible to have this relationship with them and infusions weekly and all that's going on, and not be able to tell them what's happening and to let them talk to each other without actually having any firm knowledge. So we started telling people what's happening, but we also advise them, we want it to be objective. I want you to think hard about what you're doing. Don't necessarily communicate for everything. Just do our best job of figuring out what's objectively happening and giving us that feedback, and we'll keep you updated on what we're seeing going on. And if we have an allergic reaction, we need to tell people, we've seen this, here's what would happen, here's what we did, and keep people connected in something that's so important for their lives. The phase 2 trial was successful. It was open label. All the questions, well, how do you know it's real? Well, we did the randomized phase 3 trial, and it obviously was positive. And Ryan there on the right side there is now been on the drug for 24 years. Most recently in MPS 7, we had a similar kind of thing. It's also an enzyme replacement therapy like MPS 1. In this case, it was a phase 2 open label trial, but very heterogeneous population. And it turned out some of the kids were having too much fluid, and parents could start telling you there was a problem. Now, if we had done a randomized trial, we would not figured out that they were probably getting too much fluid and they were getting kind of fluid overload, which would allow us to reduce the dilution volume of their infusions on the fly. Now, if we were in a randomized trial, we wouldn't have figured that out, and we would have not heard from them and found out what was going on and reduced the fluid instead of waiting for trouble to happen in a blinded study, then unblind them, then discover we put some kids into heart failure. So we were able to make those adjustments, and we did do a phase 3 study that also came out positive. Another program is Crysvita. This was an open label phase 2 study. Now in this case, these are kids with a bone disease due to a low phosphate. And here we could use, in fact, x-rays and blind the x-rays, but you couldn't blind if the parent sees their kid's legs straighten. It's straightening, they see it, right? And the question is, what about the other physical effects of low phosphate? When you bring their phosphate to normal, does it affect their walking otherwise? And they were telling us these kids are more active, they're feeling good. Is that placebo or is it not? Well, we also studied, for example, 6-minute walk test, which is an effort-based test and one at risk. How much effort do you put out? If you know you're on drug, would you try harder? In this case, we were testing 2 regimens. It was open label. Some kids were getting the drug every 2 weeks, some every 4 weeks. And we had the sense from them that there was a difference. And when we do the formal testing, you can see the every 2-week dosing was much better. And what was happening, their steady phosphate level was giving their muscles more ability to function. Now, the parents didn't know whether it was steady phosphorus or not, but clearly, what they were telling us is true, and when you measure it objectively in this approach, we're able to show that. Now, 6-minute walk test is one of those ones you could say is highly effort dependent. But the truth is, when we've done a lot of studies with it, you generally have not seen a large effect when people are not better. In this case, 84 meters. Maybe 10 meters, 15 meters you can see, but not that much. And so even though this is open label, you can determine that something real is happening and make a conclusion that the drug is doing something. And also, this just helped us decide between a 2-week regimen versus a 4-week regimen. So blinded studies can be objective, and you can gain the right information. Now, the last example is probably closer to Angelman is late infantile Batten's. I think it got mentioned yesterday by Allison. I was involved in that because I actually did the original work on enzyme in the CSF delivered to the brain, and no one thought it could get to the brain, and Jim was talking about the virus getting to the brain. There was a time when people thought enzyme couldn't get to the whole brain, but now we're also doing ASOs and gene therapy and everything else. So these kids, if you look at the curve there, the brown line that goes down fast, well, those kids, at like a year of age, have normal function, and within a 2-year period go to devastated neurologic function. They just crash. It's like a horrible disaster you can imagine. There's no way to do a controlled trial in this. It's an open label trial, but they used a functional scoring system, which was to gain objectivity. They have a third party neurologic scorer who's going to evaluate the kid and score based on things the kids can do. And they ran a trial compared to the natural history control using the same approach, and were able to show a very dramatic effect. Now, if this was a subtle difference in a small and slow disease, it would have been harder to have a scoring system like that not potentially be biased or not be biased enough to cause a problem. But when it was a dramatic effect, it was very doable and was approved based on this single-arm study. I think in this case, the example is a third-party observer doing the testing, and that is one of the things that we are also doing. So we believe open label studies can routinely predict what will happen. I've done it many, many times. But you need to be careful. You need to think about how to be objective. You need to work with parents about really figuring out what's going on and making sure that they're really being anchoring to things that are measurable in their minds. We like to use quantitative scores where third parties are measuring things, or we can blind the assessments. There's some things that are well known to be very difficult, like pain, some effort-based tests. And in those some of the situations, you have to be looking at large effects, otherwise you have a little harder time figuring out whether they're real or not. But I think in doing all of this, transparency, I think, is really important, and in keeping that connection with families, and it's just different from any other rare disease, any common disease area. So we need to work, the PIs and the companies and the parent need to work on being objective and doing the best they can, because we need to develop drugs that have real change, right? Not ones that are theoretically improving. So let me transition on to the second topic, which is the compassion topic. Compassion access is one of those other areas that's sort of been very problematic in history, and generally discouraged by industry, and really not encouraged by FDA either. I think the general philosophy is get drugs approved promptly, and that's the best way to get access, not through expanded access. And I would say, yeah, understood, but in rare diseases, it's a lot different. The situations are different, and seriousness of situations is there. And I remind people that we are working in the life and death business. And so we need to engage in the crises that exist in people's lives. Now, I wrote in the book because it was the most difficult period of my career was dealing with this on the MPS I enzyme therapy program. And there's a chapter in the book called Compassionate Abuse, talking about the difficulties of breaking through the standards and approach that people apply. And the truth is, it's not about ethics, it's about humanity and what you do, because it's not about saving people, it's about connecting and trying to be showing up when you need to. So let me talk through some of these examples. Now, during the Ultragenyx program, we had done a first trial, which was open label, very successful. We thought we were going to get approved. And the FDA said, well, it wasn't randomized control, so now we want you to do a phase 3 trial. Well, that meant 3 years, another $100 million. Not too much, right? And then it meant all these patients who couldn't be in the trial, who were essentially going to probably die during that 3 years. That to me was tragedy. We had some enzyme around. I thought, well, let's find some of the sickest kids in that they're not going to make it and treat some of them. And that was, created the whole crisis because no one wanted to do it because everyone was afraid of it. While I was debating this as year 2000, this little kid, he looks little, but actually he's a teenager. I think he's 16 or 17 here. He came with his mother, they're Croatian. They flew from Europe to my office in Los Angeles and showed up without an appointment, just, hi, we're here from Croatia. I want enzyme from my child. And, I sat in my room, we were talking through everything. When I left the room, the mother told my nurse, can you just give him a little enzyme while the doctor is out? Can we figure out a deal? She was an advocate for her kid. No, the truth was I couldn't get anyone in the companies to do it. Everyone was blocking us. And then I had to deal with his sister calling me, telling him that he had been found down a year or so after this. And to ask me whether pupils fixed and dilated was bad. And I said, yeah, that's bad, and he died. But I had that on me for all this time. We had enzyme, we could have treated him. He was a very smart kid. He was having respiratory breathing problems. That's why he probably collapsed. He wasn't breathing right. And I felt, what a mistake. How do I break through this? Well, the one way you break through this is you leave that company, you start another company, you set different rules. And the new rules of the new company are that we're going to deal with the crisis that goes on. We're going to take the risk if someone's really sick. And we were just getting ready with our next MPS enzyme therapy to enter the clinic. We hadn't actually treated anyone. The mom of Matthew called me and said, he's in the ICU, his pulmonary failure has progressed, his liver and spleen are very big, he can't breathe. And they have him on the ventilator because he can't breathe. And she's saying, where's that therapy you've been working on? And I realized we're now in that situation. We're going to have to watch and think about this kid losing him. And then we had people telling me, well, what if we treat this kid and he dies? What's going to happen to our drug? Well, the FDA has answered that question. They've never stopped the approval of a drug because a compassionate access patient died. It has never happened. And I had someone from FDA speak at one of my conferences. So that's just not true. And the other question was, well, if you treat one person, you have to treat everyone. What are you going to do about everyone else? I said, well, not everyone's asking. Right now we've got one. But the question I always find odd is the inability to treat everyone is an excuse for treating no one? Really? I always bring up the Titanic example. If you're in the boat and you got a spot, do you go try to save one guy in the water if you can't save 1,000? Yeah, you should. I'm going back. And that's what I talk about. The last thing they said is, can we play God with people's lives? Well, denying treatment to everyone is also playing God, a different type of God, maybe Old Testament type God. But that's God. Now, the other thing you do, you can say, no, let's get an ethicist involved. And if you want to lose common sense in anything you do in your life, get an ethicist involved. Yeah, and so in the Zolgensma story, and I like to bring this up, they brought a team of ethicists. They came up with a plan, which was, well, not enough drug, we're going to do a lottery. I said, oh, good idea. How did that go over with parents? Did I win in my life? Come on down, your kid's saved. The rest of you, you're all dead. Sorry. What? A lottery? Come on. Seriously. Right now, this kid's asking me, not everyone in the world, this kid's asking me. We gave him the enzyme, so he first ever treated, his breathing respiration improved as expected because we had the science behind what we were going to do. It saved his life. His breathing got better. He's been on treatment now for 9 years and doing really well. So that's the story. Now, we've had a kid from Malaysia ask for treatment. There was no way we're going to get, we got drug to him, but there's no way for us to figure out to commercially do that. We're trying to get a kid in Algeria. The Algerian government won't let the drug into Algeria for whatever reason, even though we're going to give it to them. But we still trying to work and treat patients where we can with MPS 7. But this is the way you have to be, right? This is the right thing. And you deal with the risk. The truth is, now here's the funniest part of this, is that the regulatory fear that treating someone, maybe they would die, it was actually flipped on its head because it turned out his data was so compelling to the FDA, they thought his data was the best data we had, better than trial data. because he was really sick and he came back from the edge and they proved the enzyme was doing something to them. So it was the exact opposite. We didn't take risk, we actually helped us. Now, in addition to that, we've had another program where it became even more critical with fatty acid oxidation defects, or LCFAOD. In this program, these are kids that have an inability to break down, create energy from food, and they can go into crisis, heart failure, muscle rupture, and severe hypoglycemia. They basically run out of energy, the tank's empty, and they can die. They get hospitalized a lot. And with this program, the treatment, we believe, had an effect on improving their energy status. And we were starting to get requests, and we knew this was going to be a problem. We have a lot of patients with FAOD who would be in crisis. But we said we're going to treat these kids because we had drug. They couldn't be in our trial because they're too sick to be in our trial, so we couldn't really enroll them. When we could enroll them, we would enroll them, but if they couldn't enroll in a trial, we had to deal with them where they were at the moment they were. And some of the kids were on heart-lung bypass in an ICU calling us for treatment. So what are you going to do? Well, the standard is there's a lot of process and time. I said, we're not going to do that because that's not being engaging in the crisis. Our view is when we set the standard, we would make a decision within 24 hours. Within 24 hours of request, we will make a decision. That's not business day. That's any day. 24 hours. And they would aim to ship drug, get drug to them within 48 hours, ship the drug to 48 hours. That means get all the paperwork, anything you need to do. Now, in some countries it's different. In the US, basically we were hitting the FDA up with all these cases. We had several dozen US, 67 patients ultimately got it. They were having muscle, heart, hypoglycemic problems, whole list of them there, they're distributed. 80% of the kids got better and survived. But 20% didn't, and there were some kids who were in multi-organ failure who ended up dying. So what happened? We submit the report, but they were very sick when we started. There was no impact on the product. None at all. What about parents? Their kids died getting your drug. They were all thankful because we got to them quickly. The kid was far gone, they knew it. It couldn't save them. But they had the closure of knowing they even got an experimental drug to their kid, and that wasn't enough. They've done everything. We had no problem with them. In fact, they were all very grateful. The truth is, about being a hero, it's not about saving people. It's about showing up. A hero, even if they died because he showed up quickly to try to save them, or in 80% of the cases, they did recover, including people who had been given last rites, heart failure resolved, came off the heart-lung bypass and went home. So it could happen. We don't get those cases. And now we have a product where all these doctors have seen this. And now when you come out with the product, their view is they know what this can do. We've been with them in the crisis. And the program's doing very well. And we have now 400 or 500 kids around the world that are getting this treatment, which I think is a great move. And this program is the right way to be. And I'm talking about it mainly because I want to change the mindset of how this can be done. Now the question everyone's thinking, oh, you're going to do Angelman computes for everything. Well, no, I didn't quite say that. Now, I will go into the Angelman program now, but I want to be clear about one thing. These are kids that are going to die, and we're in a critical situation. And we're in a situation where we had data that said that could help them. We'll talk through the Angelman program, but I think Angelman is a different situation. But it will come up a little bit later in the talk. But we have to make a good medical decision. We got to know what we're doing. We got to know we're not doing harm. We got to understand the safety profile, understand something about dosing. So I'll do now switch to this part, which will be the review of where we are in Angelman syndrome. We're doing an antisense oligonucleotide with the GTX-102. Many of you are probably aware of the program. On the bottom there on this graph, the targeting of GTX-102 is shown. It's in cluster 2, a particular place near the 5' end of the antisense message, based on the work of Scott Dindo, who is here somewhere. Where is Scott? Oh. Basically, the work showed that at this 5' end of the message, we're able to have a bigger impact on knocking down the antisense and inducing EB3 expression. So, just to be clear, we just talked about transparency. We're talking transparently. I've received some criticism for doing that. I've explained why we do what we do. We'll talk about where we are, but this is still an investigational product. We still haven't proven efficacy and safety yet. We're in the process. And open label data has its limitations. But we believe what we're seeing is real, and we think we need to continue moving forward. So we'll talk about that data, but this caveat is always true. So the program, just to summarize, and I won't go through all the history, is what we're doing right now is we're providing 4 monthly doses, dose 1, 2, 3, 4, and you can see the evaluation points. And then evaluating day 128 is a few weeks after the 4th dose, which is not very far after loading the patient with drug. We then go into an every 3-month maintenance phase. We've treated and presented data on cohorts 4 and 5 on the box on the right. I won't present 6 and 7 yet. We're just presenting today what data we presented in July with some update on safety and other factors. But these doses for these cohorts I'm showing will be at the 2 lowest doses on the rung. Now we're starting low because we had that safety event at a higher dose. So we're starting low and we're going to work our way up, monitoring safety carefully and looking at efficacy and trying to understand where do we get enough efficacy to be comfortable and still maintain safety. Some of the kids now, after the first 4 doses, if they haven't achieved a level of improvement that we consider enough, which is improvement at least 2 domains of 2+, they can titrate up to the next dose level. So those kids that started at the low doses still get a chance to get to higher doses. They're not stopped. And this is a dose titration strategy. It's part of what I prefer in the rare disease world, because if you do everything fixed and regimented, it takes you forever to get to an answer, and then all the kids that contribute to a low dose cohort get stuck at a low dose forever, and that's just not right. And so titrating is the way to get individualized dosing. And what I'll guarantee you is that no matter what, in neurology, you'll see variable doses. It's just going to be true that not everyone will get the same dose in the end, I believe. So what did we see? The CGI score, the Global Impression Scale score, this is now being conducted outside the US, at Canada and the UK. And these are different investigators scoring. And these are the 9 patients that we had data at that time. You can see that the patients, among those that respond, that they have 4 domains that are responding, and some of them, particularly 2 with the sleep and 1 with communication, had 2+ or better. We were very encouraged by this. Some people didn't respond. There are 2 patients not responding. But there's enough multi-domain responder, enough detail to what parents are describing that's responding to give us confidence. And I'll dive a little bit deeper, but this is the CGI. This is the investigator's opinion of what they're seeing at that time. It's not the latest data at this point. Now, when you dive into the objective data, which is the data that's now a third-party psychologist who is evaluating, what we were seeing in the Canada-UK patients, in the columns for the Bailey receptive and expressive communication, there's 9 patients listed vertically there. There's just 9 patients without their patient ID numbers. But the top 6 are the younger ones, and the 3 on the bottom are the older ones. You can see that there are 6 out of 9 had green scores with the asterisk. That means their change was larger than what's normally the variation of the test. Statistically different from normal variation. I would say to you, having been involved with the Bailey test in multiple MPS programs and other programs, and Ali Skriner that works with me as well, we've never seen Bailey scores really move. So these movements are something unusual and I think important. The receptive seems to be more advanced than expressive. Expressive is more complicated. I think that makes sense. I think it's encouraging. We think it's real, but it's still early and we're just at the beginning. If you look at the ORCA, and there was a lot of discussion about the ORCA yesterday, you can see that the ORCA, 2 of the patients and the young patients had statistically significant improvements in their ORCA scores. The 3 older kids also had an improvement, not quite at the significant level, but encouraging signs of some movement in the ORCA as well. I'm not going through all the verbatims on the right. Those are details that just tell you a little more about why patients are thinking what they're thinking. One domain that was really profound is sleep. Now, we all know how important sleep is, and it's particularly important if you're not getting it. But We saw in the first 5 patients, there were a couple kids with sleep problems that had a very profound effect on sleep, and we are seeing a profound effect in the kids with the worst sleep problems, and particularly 11301. That patient there has this is the Angelman severity scale for sleep. This is a quantitative scale of how much sleep they get and how their sleep character is. This patient went from a severe to a mild, a -4. This is only a 6-point scale, so you can only change that far. But this is a person who was not sleeping, waking up 3 or 4 times a night, and now sleeping 12 hours straight. And so that was very unusual. And so we've seen this pattern in sleep before, and it's really important because for the family, everyone's not sleeping because of this, and everyone then is affected the next day. But we're real encouraged by how much a profound effect on sleep we're seeing. The EEG is an area we think is important. I think there's a lot of good belief that EEG should show it. Now, before we saw some trends to improvement in delta power on the right, but on the left, it kind of looks mixed right now. It's not really showing the effect that we saw before. I think it's the dose. This is from the lowest doses now. This is not the higher doses. So I think it's just a sign we need to go up on the dose. But we'll look at the EEG as one of those factors that will help us look at the physiology of what we're doing. And I think we would expect that the delta power should go down. So the safety. Now, those efficacy deltas were from the lowest doses, but since then the patients have been titrating. We now have people getting multiple doses at the 10 to 12 range, and patients treated for over a year. We haven't seen the adverse event of epileptic weakness in anyone, or even any neurological problem of that type. So we're feeling pretty good about total 109 doses administered, 61 milligram dose accumulation. So we're feeling like we're getting a much better place of expression of dosing and without having the safety problem. We're still trying to learn what's going on, how the drug works, but I think the fact we're not recurring the safety problem is important. I think the fact we're seeing efficacy and we're now moving into a dose range of the 10 to 12, which is where we'd expect from non-human primate data to give us near maximal knockdown, is important. And as we load at those doses, we hope we'll be able to demonstrate even better on the efficacy side. Now, 2 patients from that first trial, all 5 have been unable to get drug because of FDA's restriction on that. We then provided the additional data they'd asked from us, which took time, but we weren't able to get the US open promptly, and 2 of those patients have gone to Canada to get treated and have gotten 2 or 4 doses, and they didn't have any recurrence of their problem, which is good, and they seem to be having a recurrence or improvement again that they had seen. So we're encouraged by that. But while this is going on, the crises exist, and so I got a letter from one patient from the first study, who's distraught because for 2 years they've been off the drug and they had been treated, and they sent me this letter, and they said when they got treated before, for the first time in life, the patient was able to sleep through the night, feed themselves, play with their sister, be truly connected, problem solve, follow directions, learn new skills. And now they've lost every single thing. And they're just distraught. 2 years have already passed now and we couldn't get them back. Although normally I would not have done compassionate use in a program like this, I felt like, this is one of those crises that's going on for this family, and we were having trouble getting everyone into Canada promptly enough. So we decided to tell them to send the letter to the FDA, and the FDA connected them with Dr. Barry Kravis, to do expanded access. And the FDA said yes, and the patient just got redosed. So they raised the cap, which in the US program, which I'm not talking about today, was at 2, which was arbitrary. They've raised it to 7.5, which is good, and we're submitting more data on higher doses to hopefully get the cap raised further, so we can, hope would be to open the US sites and to get the trial working across US, Canada, and the UK all together. Now, just to reiterate in bold, we're not planning a compassionate use program for Angelman. It's really not the right disease and situation. I know it's very urgent for everyone, but we really need to figure out our dosing and safety here. This is just the right way. We just can't do it. And we will be doing studies that would expand who gets treated, because I have a lot of questions about that as well. So we're continuing to load to use higher loading doses now in the next cohorts, and we'll be continuing treating the earlier cohorts at higher doses and continue to monitor them for safety. We expect an early year to expand to 20 young, 20 older patients to get more safety data. That's our expectation at this point, and hopefully in the US if the FDA allows. So that's the phase 2 study. If we can prove the dose and efficacy and get confident that we have what we need, then we'll proceed with doing a randomized placebo-controlled trial, which I know no one wants to hear, but the truth is, for Angelman syndrome in this ASO, because of the kinds of assessments, that doing one high-quality randomized trial is the right way to get proof that the drug is doing what it's doing. However, there are a lot of other subtypes of Angelman which are not in this. This is age 4 to 17 that are not included. And so we would expect to do studies in UPD, missense, in the younger and older patients in parallel studies that we would expect not to do controlled studies, but do supportive studies that we can look at and make sure we're covering the Angelman population. I'm not afraid of treating UPD. I do think actually it's going to be fine based on what we know, and we can go more into that. But that's where we are right now. I think it's encouraging. We're seeing some good effect. So far, safety's been good, but we have to keep vigilant and managing carefully. And I do believe now at the dose level we're at, we need to treat enough patients to get a real sense of the safety profile and assessment of the efficacy to be able to move to the next stage of this program. So that's the story. I have lost track of time, so I'm not sure if I'm way off the mark, but I want to thank families, for participating and for tolerating the long delays and the challenges, and certainly investigators everywhere, UK, Canada, and the US for all the support in getting this done, and Dr. Barry Kravis for all of her exceptional commitment. Genetics team, of course, have completed. Allison's still working with us, but Paula, Allison, Jennifer is here somewhere, and Scott, who has now joined us. And Scott Dindo, of course, because he's the guy that gave us the insights and inventions and put us in position maybe of doing something amazing, right, for the Angelman community. Thank you.